Our nervous system consists of two different parts. One of them is the central nervous system, which consists of our brain and a spinal cord, and the other one that sends the signals to other parts of the body is the peripheral nervous system. For example, if you think about moving your hand, first the signals generated in your brain will be transmitted to your spinal cord, and then, through your nerves, they spread into your muscle. But if something happens and these nerves are damaged, it depends on the severity of the injury. We would have a variety of problems that we should deal with. It is not only about moving our limbs, but sometimes you need your nerves that go to your lung for breathing. We know that we are not good at regeneration in general, but our nervous system is one of the worst. However, we can observe some degree of regeneration in the central and peripheral nervous system, but normally, the rate of regeneration in our peripheral nervous system is higher. But how does our peripheral nervous system regenerate naturally? To understand this, first you need to understand that we have two different kinds of cells in our nervous system. First, the neural cells, which are the basic unit of our nervous system, and they consist of a part that receives the information from other neurons, a part that processes the information, and eventually a section that sends the signal to a specific destination, which is probably a muscle or a sensory supply, such as our skin, in which you can feel a sense of touch or temperature. We have another type of cell in our nervous system called the glial cell, which is a non-neuronal cell and doesn't produce electric signals. They're responsible for the support and protection of neurons, and they have roles in regeneration after injury. Now, if our peripheral nerve is damaged, you have a problem with sending signals to your muscles. In this situation, the distal part of the axon, which is disconnected from the cell body, undergoes a famous process called Wallerian degeneration. In this process, the cell membrane of the axons breaks apart and becomes fragmented. In the next step, the specific glial cells surrounding the axon, which are called Schwann cells, start to release pro-inflammatory cytokines such as TNF-alpha and interleukin-1 alpha. These signaling molecules would recruit the macrophages to the injury site. Both macrophages and Schwann cells eat the axonal dead bodies, and after that, the macrophages release two crucial signals, interleukin-1 and PDGF, which stimulates the Schwann cells to proliferate. At the same time, fibroblasts, which I talk a lot about in previous videos, start to release two signals, NGF and IGF-1, which both of them improve survival, growth, and regeneration of neurons. At this moment, the Schwann cells start reorganizing themselves in the shape of a tunnel to guide the regenerating axons to the correct location, because it's not just about the regeneration only, but also the axon must be guided to the correct location, otherwise the regeneration would be useless. But we are not lucky all the time, and sometimes the sprouts cannot reach the right location. We can solve this problem with surgery. The gold standard treatment for nerve defects is called autologous nerve grafts, in which they borrow a piece of nerve from another intact nerve in the patient's body, and then suture it to the two ends of the nerves. But this comes with some drawbacks. One is that you should sacrifice one nerve for the other, and there's not enough source in our body to borrow tissue. And the second one is that it would require a second surgery for removal, which could cause pain and more injury to the patients. Therefore, we use a structure called nerve conduit or nerve guidance, which can facilitate nerve regeneration. However, if we use biomaterials to make our conduit for transplantation, we create an environment which has physical, chemical, and biological cues for stem cells to know how to regenerate the neural tissue. For example, in the study, they used this method for regenerating the sciatic nerve. It is the longest and widest single nerve in the human body, going from the top of the leg to the foot and the posterior aspect, and it is used in a variety of experiments all the time. In general, what they did was they made a scaffold with two different polymers, and they cultured a specific type of stem cells on this conduit. For making the scaffold, they made a conduit with an outer diameter of 3 mm, an inner diameter of 2 mm, and a length of 7 mm. Let's zoom in a little bit with a scanning electron microscopy. It has a double layer. The inner layer is made out of 50% PLA and 50% PCl with porous sponge structures, and the outer layer is made out of PLA with multi-filament fiber mesh. The important part of this scaffold is that the inner layer has a honeycomb structure with pores of 20 to 400 micrometers in diameter. And this feature is super important because the Schwann cells, which are crucial for axon regeneration, have a size of about 220 to 400 micrometers, so they can move freely, and it makes these structures ideal for peripheral nerve regeneration. Now let's talk about the cells. If you want to make fully differentiated and functional cells, we need the stem cells. In this article, they use induced pluripotent stem cells, which are different from embryonic stem cells. Both of them are pluripotent, which means they can make almost all of the cells in our body, but embryonic stem cells are separated from the blastocytes in the early embryonic stage. But the induced pluripotent stem cells are made in the lab by reprogramming the fully differentiated cells with specific signaling molecules called Yamanaka factors. In this study, they reprogrammed the human skin fibroblast cells with these 3 factors, and then they differentiated the cells into a neurosphere, which is composed of free-floating clusters of neural stem cells with the signaling molecules called fibroblast growth factor 2. Now these neural stem cells tend to differentiate into neural-like cells or glial-like cells. But if you passage the neurosphere 2 or 3 times, the cells show gliogenic differentiation and they most likely differentiated into Schwann-like cells. So they culture these neural stem cells on the conduit and then they transplant this into the mice, which have sciatic nerve defects. And you can see the nerve completely regenerated after 12 weeks. There are a lot of concepts in nerve tissue engineering that we should talk about, but I think that's enough for this video and thank you for watching.